So I'd like to thank Peter for really a very interesting and slightly embarrassing introduction. So today it's my great honor and privilege to be invited to give a talk here in this very distinguished audience. So today I'd like to share with you my journey of development as a scientist. So this is a picture of myself actually taken over 40 years ago when I was a student at St. Joseph's College, which is a Catholic school in Hong Kong. Actually, we're celebrating our 150th anniversary this year. So as a student, I am particularly interested in science, especially the science of biology. So at that time, actually, the textbook we're studying is called Biology, a Functional Approach by M.P.V. Roberts. And in this textbook, they have many pictures of famous scientists. And one of those pictures is this picture of Watson Crick standing in front of King's College Chapel. So I become very fascinated by Cambridge, how this university over the last several centuries have continuously trained generations and generations of remarkable scientists. So I've decided to actually try my luck and apply to Cambridge. So fortunately, I was accepted by Emmanuel College, which in that year was actually celebrating its 400th years anniversary. Now, so you can see that the college grounds of Emmanuel is very beautiful. And interesting, actually have many ducks. And one reason is because it has a ponds where the duck can play. So as it turned out, many years ago, there's one of a fellow of Emmanuel College was a name of Thomas Young, who's a polymath, who's interested and excelling in many subjects, from archaeology to science. So the story is that actually Thomas Young was walking along this pond and saw the ducks actually playing the water. And then you can see that the wave actually interfere with each other. And so this led Young to thought about his very famous double slit experiment, so to talk about the wave properties of light. So I think that story basically teaches me that as a scientist, we have to be very observant about our surroundings. We can actually often gain inspiration from our daily lives, as I will also illustrate to you in my own experience. Now, so in those days, if you want to do medicine in Oxford or Cambridge, they only accept you for 3 years. And after that, you have to compete and then apply for somewhere else. So in my case, after 3 years in Cambridge, I decided to maybe time to move over to the other side, to Oxford. And so I applied and was accepted by a college called Christ Church, which in terms of the size is actually the largest in Oxford. So you can see architecturally, Christ Church is really very remarkable. And one of the most remarkable features of Christ Church is, of course, the dining hall, which is literally like a museum. And of course, this dining hall was subsequently made by the Harry Potter movies into the dining hall there at the school. So this is a picture of myself as a medical student in Oxford. So as a medical student, we have to study different subjects. And one of those subjects is obstetrics. So learn about how when a woman is pregnant, she'd like to know about the health of a baby, including the genetic health. And at that time, to actually test for the chromosomes or DNA of the baby, you have to stick a very long needle into the uterus in a process called amniocentesis. But every time you do that, there's a risk that you might harm or even kill the baby. So as a result, many pregnant mothers and their families were very worried. So as a young medical student, I was thinking, is it possible I can do away with all that by inventing a simple blood test that can do on the blood of the mother? But however, thinking, if you have to do such a blood test, maybe the baby would release its cells in the bloodstream of mother. But the question is, how do you know that a cell is from a baby rather than mother? So I thought about it, and I thought, well, if the baby is a boy, then maybe I'll start to see male cells in the blood of a woman. So I persuaded a pathologist in Oxford to actually let me do some of the experiment in his lab. So we're able to actually do that. So we have a probe, which every time it sees a male cell, it has a tiny black dot. So here in this slide here, you can see a number of male cells in the blood of a pregnant mother. But then I thought, well, if every time I have to look for those cells, I have to study in a microscope, and it would be very relatively time-consuming. Is it possible I could do something more automated and faster? And then I remember as a medical student, I went to a lecture by Sir John Bell, who at that time was a young researcher, just returned to Oxford from Stanford. And he was one of the first person to actually do the polymerase chain reaction outside of the people who invented the technique. And of course, we know that polymerase chain reaction, or PCR for short, is a method to amplify DNA experimentally. So Sir John told me that he thought that this technique is going to change the world. And so I was in the audience, just like some of the students here, and I believed him. So after the lecture, I went out to him and asked him, can he teach me how to do PCR, which he very graciously did. So then I thought, well, can I try to use Y chromosome PCR to detect the male fetal cells in the mother's blood? So we did that and showed that it can work. So we published that just in a year when I graduated as a doctor. And then a few months later, The Lancet actually published an editorial called Is It a Boy? Because theoretically, you can actually use this technology to tell the sex of a baby. And then I qualified and then worked 1 year as an intern in a hospital. Actually, at that time, as a junior doctor, you have to work around 120 hours a week, like maybe some of the junior doctors here. So, but however, in my spare time, I was constantly thinking about this field because I thought I've started it and I have some responsibility to finish it. So after 1 year, I called my father up and said, well, I think that I'd like to go back to be a PhD student to work on this full time. So as you heard from the introduction, my father is also a doctor. So he was kind of worried because he was worried that I might have wasted the previous 7 years to train as a doctor, but without working as a doctor. But I convinced him, I said that after a number of years, I'm going to go back to be a doctor. So this is a picture of myself as a full-time PhD student in Oxford. You can see that I'm sort of covered up very well, almost like a terrorist. And the reason is because I'm actually using the male chromosome as a marker. So, and of course, every cell in my body has a male chromosome. So I'm trying to protect my samples from myself. Now, but sometimes life, if you do something part-time for fun, it's all good. You all see all the good things. But when you start to work on it full-time, day in, day out, suddenly you see all the problems. And this is one of those things. So even though fetal cells is present in mother's blood, but the concentration is super low. So after about 3.5 years of working on this, I cannot get a system to be worked very robustly. So at that time, basically, the research is not going so well. But in my spare time, I managed to know my wife, Alice, and so the two of us graduated together in '94 in the summer. And then I went back out to be a junior doctor. So I was trained in internal medicine first, and then later on changed into pathology or chemical pathology after watching that movies. And then 1997 came, so that was the year when Hong Kong went back to the sovereignty of China. So my wife and I thought it's time to go back home. So I joined the Chinese University of Hong Kong. So this is our campus in the northeastern part of Hong Kong, which is a very beautiful Tolo Harbor. Now, interestingly, about 3 months that I'm supposed to leave Oxford to come to Hong Kong, in this issue of the journal, Nature Medicine, there were 2 papers. They say that in patients with cancer, you can find cell-free cancer DNA in the plasma and serum of a patient. And then suddenly I have a strange thought because I thought that a baby living inside mother is actually a little bit similar to a cancer growing in a patient. So maybe for the previous 8 years, I was looking in the wrong place because for the previous 8 years, I was trying to look for fetal cells in mother's blood. But maybe I should be looking at the cell-free part in this fluid called plasma and try to see if I can see cell-free fetal DNA. But then the problem is that at that time, I was just about to move from Oxford back to Hong Kong. I don't have a grant. I don't have a lot of resources. And I've never actually handled plasma before. So how can I do it? And then I remember, even though I was living in the UK for 13 years, I never liked UK food. So being a Chinese person, so sometimes I actually go and cook noodles in my room, right? So to cook noodles, what you do is that you boil some water and put your noodles in, and a few minutes later, you have your meal. So what I did is that I thought, well, how about if I boil the blood plasma for a few minutes and take a few drops of that boiled soup to be tested? And you probably thought that you're crazy. But sometimes when you don't have resources, you have to do crazy things. And this is what happened. So when I did that with 10 microliter of boiled plasma, I was able to use PCR to detect a Y chromosome in some women. So you can see those with the white line. And some of the other women, they don't have this white line. And later on, when I wait for the women to deliver, I find out all the ones with the white signals all have baby boys, and all the ones without signals all have baby girls. So this is the first demonstration of the presence of self-revealed DNA in maternal plasma and serum. And then I tried to measure the concentration of this DNA. And I found that actually this DNA is detectable very early on, from the 7th week of pregnancy. And actually by the 10th week, the concentration reaches 15%, which is amazing because by 10 weeks, the mother is still huge, the baby is tiny, and yet the baby is working so hard to push 15% of the DNA into mother's blood. And then after the baby is born, within 2 hours, the DNA is gone. So then I thought, well, maybe I can try to use this method to detect some diseases. Now, the number one reason why pregnant women go for prenatal testing is because they're worried the baby might have chromosome disorders like Down syndrome. Now, Down syndrome is caused by abnormal number of chromosomes. Now, in a healthy human cells, you have 23 pairs of chromosomes. So chromosome 1 is the longest, 2 is a bit shorter. So in Down syndrome, number 21, which is a very small chromosome, instead of 1 pair, we have 3 copies. And normally, to detect this condition, you have to take a cell from a baby, and then you probe the cell, and then you ask, how many chromosomes 21 this cell has? So in this case, you see 3 red chromosome 21 signals. So the baby have Down syndrome or trisomy 21. But however, we have a problem because in my field, I'm not looking at cells. I'm looking at DNA swimming outside cells. So the question is, how can you use a signal which is outside of cell to deduce how many chromosomes are inside a cell? It's almost like you try to find how many people in this room, but you can only do that by looking at the number of people outside the room. It's a bit challenging. So it actually took us 10 years to work it out. But the final answer seems very simple. Let's say that you have a wallet. Sometimes you have, say, 4 coins, and some other times you only have 3 coins. And I want to know how many coins you have in your pocket today. Now, one way is to ask you to hand me your wallet, but that's like invasive prenatal testing, very intrusive. Now, another way is to ask you to stand on a balance. Now, theoretically, the day when you have more money, you should be a little bit heavier. But your balance has to be super accurate, right? It can differentiate, let's say, 60 kilogram versus 60.01 kilogram. So what it means is that we have to develop a very accurate way to quantify DNA. So as I told you just now, most people quantify DNA or detect DNA by PCR. But PCR is a doubling process. So it's very good to tell the difference between 100 and 200. But it's not very good to tell the difference between 100 and 105. So we calculate that to make this work, the precision has to be like 5%. And furthermore, inside mother's blood, most of the DNA is from mother, like the red signal, with very little baby's DNA, which is the yellow signal. So normally, if you do everything together, the mother signal is going to overwhelm the baby signal. So we thought, well, how about if I don't analyze everything together, I distribute it into many, many reaction wells, so that one well will only have one molecule or no molecule. So in this sort of situation, the baby's DNA can be analyzed separately on its own, and you can make the test to be as precise as you want. You just work harder, have more reactions. And so we did this, and in 2007, we published for the first time that you can actually do a non-invasive test from the mother's blood to tell the babies have Down syndrome or not. So we published this, but strangely, within a few weeks after this publication, there was another paper from another group, and you can see from the title, it's almost exactly identical. And furthermore, the two papers were submitted on the same day, which is incredible. So that means that we have competition. But on the other hand, the good thing is that you now know that two independent groups have arrived at the same thing. So probably what we have talked about is correct. Now, this method, even though it works, it has a bit of a problem. And what is the problem? The problem is that to analyze something by PCR, you have to design a primer to a gene, let's say on chromosome 21. But on chromosome 21, there's actually 150 genes. So by targeting on one, you're wasting the information on the other 149. And that is bad because we already don't have a lot of molecules and you're wasting information. So I'm thinking, is it possible I could develop a method which will allow me to maximize the use of information in the mouse blood? So at that time, a new technology was just coming out. It's called next generation sequencing. But most people, they use the technology at that time, we use it qualitatively to ask if a tumor has a particular mutation or has a particular virus got some variations. But what we're thinking about is, can I use this method quantitatively and in a random sequencing manner? So I was imagining, say, in mother's blood, there's many DNA circulating around. I sequence each molecule, and then because I know the human genome sequence, I can map each molecule to the chromosome from which it comes from. And in the bottom here, I can just tally to calculate the representation of different chromosomes, whether the chromosome 21 has the same representation as the other chromosome. Now, interestingly, when I did this, I find that it has to be very good. So we try, for example, here you can see the pregnancy with Down syndrome babies. The male ones are in green, the red ones are in female, and the signal is very high compared with the normal pregnancy, which is in blue and orange. I try 28 cases. Half of them have Down syndrome, half normal, have 100% accuracy. So very excited, and then we published this paper in a year after. But very interesting, actually, within a few weeks of this paper, the other guys published the paper. And then I started to wonder, so who is this person, right? So I looked at the last author. I saw the name is called Stephen Quick. And I looked at his website and find that he's a professor in Stanford. Now, interestingly, he also got his PhD from Oxford, and actually in the same year as well. So my wife is a physicist, so I asked my wife, do you know this person? And she said no, because Quick at that time was a theoretical physicist. My wife is empirical physics, so they didn't know. But my wife very intuitively decided to bring back this photo, and then this is Steve Quick. So it's unbelievable. It's almost like today, right? The person sitting next to you is going to compete with you in 20 years from now. So maybe you should all take a selfie together. And not only that, actually, and a few years later, because of this work, I was fortunate enough to be elected into U.S. National Academy of Science. And on the day when the result came out, I saw my name, low, of course I'm very happy, and looking a bit down, and I saw his name. So three of us decided to take a picture together in the NAS. But as you see from this photo, it looks like Stanford is a very stressful place compared with Hong Kong. But anyway, so now we have this prototype method which seems to work. So the next question is, if I want to push it into a clinical use, I have to do clinical trials. So I spent the next 3 years doing that. And this is what happens. So we find that by using this technology, I can detect Down syndrome with an accuracy of 99.7%. So we thought, well, we're ready. So this technology actually is ready to be used in a clinic. But I can't do that in the university. I have to work with some companies. And of course, if you want to work with companies, the first thing they'll ask you is, do you have a patent? So for the last 20-odd years, my group has been sort of diligently filing some patents to protect this technology. But when I first started, I didn't have a lot of experience. So I remember actually the very first patent I have in this field, our university licensed it to a company in the UK. And that company actually sat on this technology because they didn't realize the significance of it. And 3 years later, they thought it was rubbish. So they just gave it back to the university. And of course, that turned out to be a very big mistake for the company because this patent became very valuable. So to the young people in the audience today, I think that in the future, if you invent something big, I would urge you to open your own company rather than to license it out because at least you have a control about what you want to do. Now, okay, so after the intellectual property came back to university, I have to find some other people who is interested. So I remember I actually went to Thailand for a meeting, and I met this gentleman who's Charles Cantor from Columbia. And then he actually, at that time, he was also the scientific officer of a company called Sequenom. And so we start talking and collaborating, and eventually actually Sequenom licensed our technology. And of course, in the outside, you can imagine in Stanford, the professor have many commercialization experience. So Steve Quick actually licensed the technology to Prenatal, which was then bought by Illumina for about $600 million US. And now you can imagine, so Sequenom has my IP and Illumina has his. And whenever companies have IP, unfortunately, they use it like weapons, right? So they start to fight a series of battles. And I remember there's 4 lawsuits, and then we won 3 of them, and they won 1. So eventually, the 2 sides decide to settle. And so we pool all the patents together so that anyone who is interested in the field can just subscribe to this patent pool and then gain access to the technology. So this technology is called NIPT, so non-invasive prenatal testing. It is now the standard of care in many countries, and over 100 million tests have been done now. Now, basically, with this, it seems that my initial dream as a medical student has been achieved. But then I thought, well, I'm still kind of young enough that I'm not retiring yet, right? So I was thinking, what else can I do? So if you're trying to push this technology to the extreme, what would it be? So I'm thinking, well, if you push it to the extreme, you have to ask yourself, can you sequence the whole genome of the baby this way? But this is quite a difficult problem to solve because the genome, of course, is 6 billion base pair, right? And that's fragmented into millions of pieces. And then also bits of it is from mother and bits of it is from baby, and they are related and mixed together. So how can you do that? So I was thinking, can I invent an algorithm to solve that? So one solution to this question. But I could not think of a way to do it. And I remember until one day, I went to see this movie with my wife. It's actually one of the first 3D movie in which the first 10 minutes is in 3D. So I remember I was sitting in a cinema like here, put on my 3D glasses and waiting for a movie to start. And then suddenly I saw the Harry Potter logo flying towards me. And then my eyes was caught by this H in the Harry bit, which in 3D actually looks to me like a pair of chromosomes. And then I was thinking, well, for every pair of chromosomes that baby has got, it has 1 copy from a father and 1 copy from a mother. So maybe my mistake is that there's no one algorithm. You need 2 algorithms to do this. So what is the 2 algorithms? So let's imagine you look at, let's say this picture is a father's genome, and this one is a mother's genome. The baby will have 1 half from father and the other half from mother. So now, if you look in the mother's blood, there's a lot of mother's genome floating around, and you're trying to deduce the fetal genome, which is in yellow. So now, algorithm 1, to crack the father side of the baby's genome. So you look at the father's genome, and then you look at the mother's genome. And then you try to find things which is only present in the father's side, but absent in the mother's side, but which is easy to find, like the flower. And then go into the mother's blood, and then try to hunt for that flower. So every time you find a flower, there's a little bit of the stuff that the baby has inherited from mother. And then you zip all those flowers together, then out come the father's half of a baby genome. So now, how about the other algorithm, the mother half? The mother half is a bit more difficult because the baby's DNA is swimming inside the mother's blood, where there's a lot of mother's genome. So you can imagine if a mother gives some of the genetic material to the baby will have it and the mother will have it too. So how can you do it? So we have to think of something cleverer. So I can imagine, let's say if you look at the mother's genome, the left-hand half and the right-hand half. should normally be present in a ratio of 1:1, just like a pair of gloves. Left-hand glove and right-hand glove is 1:1. But now let's say the mother inherits the right-hand half to the baby, and the baby releases it back in the mother's blood. So inside mother's blood, the right-hand half should be more than left-hand half by a little bit. So basically what you do is go into mother's blood and you count the two halves. So the half which is in excess is a half inherited by the baby, and you do it 1 million times. So eventually we did this, and so this was published in Science Translation Medicine. And then about 18 months later, the two US group replicated this result, of course, including our friend in Stanford. Okay, now you can see how we developed this field, right? So originally trying to work on the fetal side, but then I was inspired by the cancer side to look for cell-free fetal DNA. But now the fetal side seems to be working well, so we thought, well, maybe we can do the same thing for cancer. So we started with a cancer called nasopharyngeal cancer, which is a common cancer in the head and neck region, common in the southern part of China. Now, for example, a Cantonese man like myself, I have a lifetime risk of having this cancer about 1 in 39. Now, one reason why I choose this cancer is because, like for the baby, I use the Y chromosome as my first marker. For cancer, you also need to have some special type of DNA as a marker. And luckily for this cancer, we have that because this cancer is associated with a virus called Epstein-Barr virus, EBV. So I was thinking, maybe in the course of its biology, the cancer cell will release this virus DNA in the bloodstream. Now I remember when I first thought about this idea, I was talking with some of the clinicians in my hospital, and many of them laughed at me because they thought that most of us are chronic EBV carrier. So if you do a test like this, you're going to have a lot of false positives. But I decided to try it anyway. And this is the result. You can see on the left-hand side is the people with this cancer. And the blood is stuffed with this virus DNA. The right-hand side are normal individuals. And then we thought, well, can I use this technology for early detection of this cancer? And we think it's worth doing now because here is a survival graph. So every time somebody passes away, the graph will go down a little bit. Now, for example, in the top there, if you discover this cancer in stage 1, over long term, 90% of people survive. But you wait until stage 4 and treat, that with the bottom graph, over long term, 40% of people die. But despite the relatively good healthcare system in Hong Kong, 76% of this cancer only discover late in stage 3 and stage 4. So we want to do something about that. So we did a large-scale screening study in Hong Kong in which we studied over 20,000 subjects using this test. And in the end, we find 34 cases of cancer. Now this number, 34, is very interesting because according to government statistics, every 100,000 people in Hong Kong, there are about 35 cases of this cancer. And so if you look at 20,000 people, theoretically, you should only have 7 cases of cancer. But here, I got 34. So why is that? Because we're thinking that our technology is able to pull out a cancer which is going to present over the next 4 or 5 years. Now, if that's the case, the cancer we obtain should be very early. Now, for example, if you don't use this technology, then most of the cases in stage 3 and stage 4, so the orange and red signal. But now, if you use our test, then most of the cases become stage 1 and stage 2. And also, because they're so early, of the 34 cases, only 1 die. The rest are cured, the top line. But if you don't use this technology, then over long term, about 40% of them die. So basically, mortality has reduced 10 times. So this work was published in New England Journal of Medicine in 2017. But this is only the first step. So we want to actually now generalize this technology from this head and neck cancer into other cancer types. But of course, the problem is that not every cancer is associated with a virus. So initially, we start with thinking, well, can I detect genetic mutations? But then we find there's difficulty because in our genome, we have 6 billion base pairs, 3 billion for father, 3 billion for mother. And most cancer just have a few thousand mutations or maybe 30,000 mutations. And then to search for that is difficult because you're searching 1 in a million base pair, and you don't even know whether the patient have cancer or not. So after trying genetic approaches for a while, we decided to move to epigenetics. So epigenetics is when you do not have a change in DNA sequence, but you have some modifications of that sequence, like formatting changes. It's like when you have a Word document, you can have different fonts, you can have different italics, color, underline, etc. So we're thinking, can we use an epigenetic approach to tell if cancer is present? So we developed this in 2013, and basically we find that with one test, I can detect multiple types of cancer. And furthermore, if you tell people you have cancer, the next question they're going to ask you is, where is the cancer? But for the epigenetic approach, we can answer the question as well, because even though the different cells in my body have the same genome sequence, but depends on the organs, they are formatted differently. So by using the same test, I can tell you where is the cancer. So this time, with more experience, I decided to start my own company called Serena, which stands for circulating nucleic acid. So basically, we're trying to develop products which can detect cancer early and allow us to do something about that. But interesting, at the same time, when I founded Serena, another company in the Silicon Valley was founded called Grail. And you can see the mission is to detect cancer early when it can be cured. So basically, Serena and Grail are moving in the same directions. So from my previous experience, you know this will end up in lawsuits, right? Every time in lawsuit, you spend years and millions of dollars. So this time, a little bit wiser, so we start talking with Grail. And eventually in 2017, we merged together, and I became scientific co-founder of Grail, and then they use our intellectual property. So this is actually at a party celebrating a merger. The background here is the NBA Finals in San Francisco. So you may recall that year is the Golden State Warrior won. So when we book this room, we don't even know that they're going to win, but we try it anyway. So the guy next to me is the founding CEO of Grail, a guy called Jeff Huber. He's the person who brought us Google Maps. And then he decided to actually dedicate his knowledge and skills into cancer testing. So eventually, after a few years of work, Grail basically used our IP to develop this test called the Gallery test, which is a test which can detect 50 types of cancer and tell you where it is. And this test was chosen to be one of the best inventions by Time Magazine 2022. And of course, you say, well, how good is this test? Now, for example, in this particular paper published in 2023, they look at over 6,000 individuals with a median age of 62%. And they find that if the Grail test is positive, the chance of this person having cancer at that moment is over 75%. If it's negative, the chance of that person not having cancer is over 97%. And the accuracy of telling you where is the cancer is 85%. So those are pretty promising. But however, it does have some weakness. The weakness is for stage 1 cancer, the detection rate is only about 24%. Stage 2 is 57%, stage 3 is 85%, stage 4 is 95%. But however, I would say that if I unfortunately to have cancer, I would surely like to do a test like that. So at least if I'm fortunate enough to be in the 24% group, at least we can do something about that. But of course, it's still early days because we need to enhance the sensitivity of this test for early stage cancer. We also need to demonstrate survival benefits like what we've done for this nasopharyngeal cancer. And also currently the test is selling for something like over $900 US dollars in the US. And for a country like China, this will be too expensive. So we're now working hard to develop a second generation test, which will try to do it cheaper. So finally, in summary, hope I've convinced you that it is actually rewarding to be a clinician scientist, because if I just work as a medical doctor, even though if I work very hard, so every day I can only serve maybe, let's say, a few dozen patients. But now as a clinician scientist, if you invent something useful, maybe you can help millions of people every year. And also as a scientist, we have to take inspiration from daily lives. Like for my case, cooking noodles, or even weighing myself, I have some new ideas, or watching movies. And also another very interesting thing is that pregnancy and cancer are very different things, but however, there seems to have a connection between those two things. If you can see that link, sometimes this will lead to breakthrough. And also, it is also very important to care about your intellectual properties, because if you don't protect your intellectual properties, then they will reduce the chance that a company might commercialize it, which will then reduce the chance of you directly benefit patient. So I think there's a synergy between academic research and the commercialization. And finally, of course, research is not just one person. We're talking about a whole team collaborating over many years. So I'd like to thank my team for working with me over the last 20 years. So thank you very much for your time.
Dennis, that was amazing. We have the privilege to see how you are writing the history in medicine today. That's amazing. Thank you so much for that. So, this is time for questions. There is one over there, and after... First, second, third.
Ricky Steuermann from Mariel University, and I'm a genetic counselor, so it's going to be very practical. NIPT, as a national program, I think has made its way only to a few countries, and I would like you to address the issue there. And the second question is how far you think we are from doing more than just a few trisomies and microdeletions and moving on to a full genome, because as a geneticist with the chromosomal microarrays and the whole exomes that we're doing extensively in Israel, and we're already even moving to whole genomes in the clinical practice, not just research. So how far before we can do that in a non-invasive pattern?
Thank you very much. So the first question, if I hear correctly, is about accessibility, right, to the lower income places, right? Yeah. Right. So I think part of that is that of cost. Now, for example, like in Hong Kong, if you do it in the government-supported program, then you need to complete other cheaper tests first. If those tests are positive, then you go for this new test. But in the private market, then many women already, they pay themselves and they access to the new technology. So ultimately, I think that probably the cost of access, like from sequencing, will help that. I mean, I can see, for example, like some of the patents from Illumina has already expired, and now there are actually some new sequencing company in which the sequencer is much cheaper, right? So I can see that, for example, there's some sequencer from mainland China, you're talking about maybe 10 or 20,000 euros for that sort of range. So I think this will help. So the second question about the spectrum of diagnostic diseases which can apply, I think that probably with an increase in the search space, with more disease that you're looking for, then the specificity of tests become more important. So I think that you need to add specificity of tests. And so in this regard, there's one area that I'm working on. It's called fragmentomics. Is the fragmentation pattern will also give you specificity. So we're hoping that that will help as well. Thank you.
Thank you very much. Questions from there?
All right. So as you have mentioned previously, you have encountered many difficulties during your research. So my question is, what was your motivation to keep on going and not giving up?
So thank you very much. So I think that my philosophy is that you should really keep no stones unturned. So for example, I showed you just now that we arrived in this sequencing test, but actually I've tried many other ways. So I probably tried close to 10 different ways and then find this one to work. But even though the other tests did not work at that juncture, but still you learn something about them. You know, there's a new concept to be made. And also interesting, one of the tests that we tried at that juncture was an epigenetic test, which didn't succeed at that juncture, but later on became the cancer test. So sometimes I think that don't just have your eyes on the ultimate answer. I think the process is important. And also that's why to be a scientist, you really have to enjoy that process. Yeah. Thank you.
Thank you. I think there were a question there. Thank you. The- Could you please stand up? It makes easier for the mic. Yes, thank you.
Thank you for your presentation. My question would be, well, I actually have two questions. You mentioned when you found that you have competition for the first time, you didn't want to work with them, you wanted to compete with the competition. Do you think it would have been better to work together with them? And my other question is, what is your advice for young researchers? How can we handle competition?
Yeah, I think for collaboration, of course, you have to have the right time, and also it helps if you already know that person, right, in conferences, etc. So I think in that situation, we probably don't have that privilege at that juncture, so that's why we compete. But sometimes, of course, competing, it does drive work faster as well, right, because both sides want to win, and then they do it. And also because you're competing independently, then your approach might be slightly different, which also would help to show the well more than one way to do that. And your second question?
My second question was, do you have any advice for young researchers at the moment, how to handle stress and how to handle competition altogether?
I think to handle stress, I think I kind of can compartmentalize my brain. So for example, what I think one should not do is that if you have something stressful, to constantly ruminate about that stress throughout your 24 hours. So for me, for example, if this is something that I try to crack, I will devote this time to think about it. And then after that, I can do some other thing else. If doing the other things, you don't think about the first one. Otherwise, you just constantly, stressing yourself. Thank you.
Thank you. And we have time for one more question.
Peter,
Eva.
So I think the Grail or gallery test is really something which gives the hope for detect cancer as soon as possible. And as I remember, as you said, the basic problem that actually the sensitivity is not good enough. But as I remember, you can detect with this test the pancreatic cancer at a very early stage. And this is a deadly cancer which recognized usually very late. So even if you have to develop this technique for higher sensitivity, cannot be used already for this type of cancer to make a general test or test specifically for certain type of cancer like the pancreatic cancer.
Thank you. Now, so the approach I mentioned to you, such as by Grail, is a multi-cancer based approach, and in which pancreatic cancer is one of the targets. And of course, you can do the same thing just for one type of cancer. But I suspect probably pancreas cancer, of course, is a devastating cancer, but it may not be high enough prevalence for you just to develop just one test for this cancer. If I was doing it, I probably would do multiple types of GI cancer in one go. Yes. Thank you.